scientists have achieved a remarkable feat by cultivating a human embryo model that closely resembles a 14-day-old embryo, all without utilizing sperm, eggs, or a womb. This groundbreaking endeavor, conducted by a team at the Weizmann Institute, involved the use of stem cells to craft the embryo model, which bears a striking resemblance to a genuine early-stage human embryo and even exhibited hormone release that led to a positive pregnancy test result in a laboratory setting. The primary goal behind these embryo models is to offer a morally sound approach to comprehending the initial phases of human life. The first few weeks following fertilization are marked by profound transformations, as a cluster of undifferentiated cells gradually evolves into something recognizable in a fetal ultrasound. This critical period is a significant contributor to miscarriages and birth defects but remains shrouded in mystery due to limited scientific knowledge. Professor Jacob Hanna, a researcher from the Weizmann Institute of Science, emphasized the challenge of this early developmental stage, stating, "It's a black box, and that's not a cliché. Our knowledge is very limited. Embryo research is fraught with legal, ethical, and technical complexities, but a burgeoning field seeks to emulate the natural progression of embryo development." The Weizmann Institute study, published in the journal Nature, claims to have created the first complete embryo model, replicating all the essential structures that emerge during the early stages of embryonic development. Rather than relying on sperm and eggs, the researchers initiated the process with naive stem cells that were reprogrammed to possess the potential to differentiate into any tissue type within the body. These stem cells were then directed through chemical cues to differentiate into 4 distinct cell types found in early human embryos: epiblast cells, which give rise to the embryo or fetus; trophoblast cells, which become the placenta; hypoblast cells, which form the supportive yolk sac; and extraembryonic mesoderm cells. A precise combination of 120 of these cells was mixed together, and then the scientists observed that approximately 1% of them spontaneously organized into a structure bearing resemblance to a human embryo, albeit not identical. Professor Hanna marveled at the process, saying, "I give great credit to the cells. You have to bring the right mix and have the right environment, and it just takes off." These synthetic embryo models were allowed to grow and develop until they reached a stage comparable to a 14-day-old embryo, which is the legal limit for embryo research in many countries. Professor Hanna, in a passionate discussion during a late-night video call, offered a 3D tour of the embryo model's intricate architecture. He highlighted the trophoblast enveloping the embryo, which typically forms the placenta, as well as the cavities, known as lacuna, that fill with the mother's blood to provide nutrients to the developing baby. Additionally, he showcased the yolk sac, which performs some functions of the liver and kidneys, and the bilaminar embryonic disc, a key characteristic of this embryonic stage. These embryo models hold the promise of shedding light on how different cell types emerge, observing the earliest stages of organ development, and gaining insights into inherited or genetic diseases. Already, the research has revealed that other parts of the embryo do not develop unless early placenta cells surround them. There is even speculation about the potential to enhance in vitro fertilization, IVF, success rates by understanding why certain embryos fail or by using the models to assess the safety of medications during pregnancy. While Professor Robin Lovell-Badge, a researcher specializing in embryo development at the Francis Crick Institute, praised the embryo models as pretty good and pretty normal, he also noted the need to address the current 99% failure rate. Understanding miscarriage or infertility would be challenging if the model does not successfully self-assemble most of the time. Furthermore, this research raises the intriguing question of whether it is ethically permissible to mimic embryo development beyond the 14-day stage. In many countries, such research remains legal as embryo models are legally distinct from actual embryos. However, the closer these models come to replicating genuine embryos, the more ethical dilemmas arise regarding their regulation and treatment. Professor Alfonso Martinez Arias of Pompeu Fabra University praised this research as a most important piece of research for faithfully reconstructing the complete structure of a human embryo from stem cells, thereby enabling studies of the events leading to the formation of the human body plan. It is vital to emphasize that achieving a successful pregnancy using these embryo models would be both unethical and impossible, as assembling 120 cells goes beyond the point at which an embryo could implant into the uterine lining.